Hello. Good evening, everybody. And I thank Dr. Srinandan for his nice introduction. So my talk is titled about the yin and yang of viruses. So as Dr. Srinandan introduced, I'm one of the my lab is one of the participating lab in the Center for Research in Infectious Diseases, CRID, which is one of the centers of our School of Chemical and Biotechnology of SASTRA University. And my interests are viruses, especially I do work with HIV. We work on targeted drug delivery for HIV. We screen novel latency reversal agents for HIV. We also validate and screen for novel antiretroviral drugs from different libraries and phytochemicals. And also my interests are on studying the epigenetics behind the HIV infection and its latency establishment and reversal. So with this background and my experiences, first-hand experiences in working with viruses, I also give courses in our university for graduate and undergraduate students on virology, nanotoxicology, and biology for engineers and immunology, and some practical courses in microbiology. That's my teaching background. Here, I would like to add some comments because based on the responses, it's mostly almost 80% of them are students, graduate, undergraduate, and PhD students, and few are from a non-scientific background also have joined the meeting. So I make this presentation as a platform to introduce viruses, and this is the best time to do that, I hope, because we have a prevailing situation now that everybody knows at least one virus name, that is coronavirus. And we know, we do speak in common terms about the virus, and we speak about its virulence, we speak about its incubation time in the body, and we speak about the drugs, whether it is available or not, and we are worrying about whether can we have a vaccine or a specific drug against coronavirus anytime soon. So there is more, and it's a perfect time. There's a more talk on viruses and care about viruses now. And this is a perfect time to introduce the whole story of viruses. So let us listen to the story of viruses. It's going to be very basic, broad, introducing many aspects of virology. So I have made my presentations in this order, inciting a curiosity about what is viruses, and then we'll go into the history and social history of viruses, and we will go and ask these questions that are unsettling, and it actually divides the scientific community into two: where these viruses came from where, and are they alive? How do you understand the viruses because of these peculiar characteristics? And then we will see what is this yin and yang that is in the title, and how the virus is going to be presented in this yin and yang concept of Taoistic philosophic view. So I would like to ask or start with some astounding facts about some organisms. Which organism global dry biomass is 230 times to 700 times higher than human biomass? Which infectious organism wiped out almost half of the Europe's population in the 14th century? Which organism could almost same or slightly more than the total number of cells of humans? It's known that it's 3 into 10 power 13 cells in the human body. I think we are made up of so many cells. There are organisms within our body, they are almost like this, or they are just above our total volume of our own cells. We have so much of them. What is it? Of course, obviously, we think that this talk is titled on viruses, so the answer should be viruses. But no, it's all bacteria. It's nothing about viruses. Okay, so let's see some interesting quote about viruses. There are beautiful quotes about bacteria, how they are useful for us, how they are in our stomach, the microbiome, how they are important for us, how bacteria are doing fantastic job in food industry, leather industry, antibiotic, pharmaceutical industry, and so many other ways. Is there anything nice to say about viruses? Let's see at least one quote. Virus, a piece of bad news wrapped in a protein coat. Sir Peter Medaver said this. It's not again anything interesting or inducing any curiosity about virus. So let us leave this off. Now let's go into the real story of the viruses. I would like to add some prelude to their discovery because it took a really hard time for people to understand those organisms that are not visible with our naked eye. Because it all started with a prevailing understanding of what is life and how new life forms are produced. Once in the whole Europe, there was this understanding. It was almost considered as a scientific fact that's called a spontaneous generation theory. For more than 2 millennia, even before the time of Aristotle, even Aristotle proposed this. From even before the time of Aristotle, for more than 2 millennia, this spontaneous generation theory was considered as a scientific fact. What does it mean? It says that the living creatures could arise from non-living matters, meaning from a laptop, a rat can be produced, or with 2 bricks and few leaves, and if you keep it under sunlight, you can produce scorpions. Yes, there were really protocols like that. People followed it, and they believed yes, it is. For example, the maggots that are produced in this rotten meat or meat that is getting rotten, they said that, see, this meat was just a meat, and now you see after 1 or 2 days that now all a hell lot of maggots are produced. What does it mean? It's a meat from a cow or from some other animal. Where does these maggots are coming from? So there is no relation between the one organism and another organism. Anything can come from something. If there is just air, that's all. It's no matter. You can have life forms spontaneously generated. Life. They even thought that formation of organisms without descendant of similar organisms. For example, from the meat of a cow, you can have different insects or something. They know these 2 ideas are called as heterogeneistic theory and abiogenesis. Heterogenesis and abiogenesis. From many things can arise. From even non-living things, living things can come up. So this theory was prevailing for more than 2 millennia. As a scientific fact, people thought that this is scientific because it sounded kind of a rational for them. Calling, because the religious climate at that time, it meant, or it advised people, or it teaches people that only a life can give rise to another life. That's it. There is nothing else. Whatever you see as a life comes from another life. That's all. So, calling the creatures can be spontaneously generated. You don't need to have a life force to produce another life. It sounded kind of rational and atheistic and scientific for them. So many believed in that also. In 17th century, first time, a scientist named Francisco Redi, he first challenged this spontaneous generation theory. He simply sealed the meat in a container, airtight container, and then, after a few days he opened it. There was no maggots. So he said that I proved, because if you are closing something, and then those maggots are not formed from the meat, rotten meat. So it's proof for or against the spontaneous generation theory. In the same century, we should remind about the discovery of, the invention of designing of microscope by Antony von Leeuwenhoek. And he observed microscopic organisms, microscopic life. And in 1677 itself, it was fully acknowledged by Royal Society. I need to stress this. It was fully acknowledged only at that time, because he started to observe small creatures in water, in dust mites, anything. But people are the Royal Society again. That's because of the mindset and the climate, cultural climate at that time. When he showed minute details of an ant or some other minute animals or creatures, insects, they were happy to see, wow, that's a beautiful technology. It shows the minute details of an organism. But when he showed that in water, what we cannot see, like an ant or anything, there are organisms that are not visible to your eyes. Initially, he was thought, or he was considered, or he was not accepted. He was considered as he's speaking nonsense, or it was not truth. Nobody could believe it. And then he had to prove again and again. And finally, Royal Society, they accepted his observations of microscopic organisms. Why I'm stressing this at this point is, this has a direct relevance to refute this spontaneous generation theory. Lots of scientists in the continuing centuries, because it took 3 centuries finally to convince the people and the scientific community that spontaneous generation theory is wrong. 18th century, John Needham and Lazzaro Spallanzani, they did some experiments, and they again put forth their ideas to refute the spontaneous generation theory. In 19th century, Charles Cagignard, Theodore Schwann, these are the scientists. Again, they were doing some more experiments to prove them. It's all starting from Francisco Redi, and even before Francisco Redi, some challenges were taken by the scientists to prove or disprove spontaneous generation theory. These 18th and 19th century scientists, I'm just mentioning the important names here. What they wanted to do is, or try to do is, they are just doing the same experiment done by Francisco Redi or by Lazzaro Spallanzani. They are improving that experiments. For example, Lazzaro Spallanzani, he boiled some broth. If you keep the broth open, there will be some growth in it. It will turn muddy. But if you boil it, and airtight, close it airtight, and even after a few days, nothing will grow in that. But the people from the spontaneous generation theory, they would argue, no, it's wrong, because you are not letting the air to go in. Air is one of the important factors to breathe into something to make it as a life. So you are closing air, that's nothing is growing. So they didn't accept it. What? Finally, in the 19th century, almost in 1859, the great Louis Pasteur and his another scientist called Tyndall, they started one. They actually did the same experiment done by Lazzaro Spallanzani. Even Schwann, in the previous slide I showed his name, Schwann, Dr. Schwann, he also said that using the microscope developed by Anton von Leeuwenhoek, he saw that broths are containing some spore-like structures. Actually, it was some yeast. It is fermenting it. So people, they didn't believe it, because they didn't consider that there can be some invisible life in the air that grows in this nutrient media. They didn't believe it. They didn't accept it. What Pasteur did is a very simplistic and a nice experiment. He did this Schwann neck flask experiment. He added some nutrient broth. He completely boiled it, and also the top was covered with a bent neck glass tube. The glass tube was also heated, so that the air that comes through the glass tube is completely hot, and if there is any dust and the germs sticking on this dust, is all completely killed. So he made 2 sets of tube. One tube, he kept it as it is. On the second tube, he broke the neck. He opened it for the fresh air to come. After a few days, the fresh air, where in those flasks where the fresh air went, there again something started growing. But in the other one, there was no growth, because the neck of the Schwann neck glass tube is bent like that, so that any air coming, already the air is completely killed, or clean, sterile, and any air, if that comes, most of the dust that sticks in the pathways or in the surface of the bending glass, so nothing gets inside. within few days, nothing could grow there. So this was finally accepted. Again, there are some funny reasons why Pasteur and Tyndall and their experiments were accepted. Because at that time there was another scientist. He was also a director of a natural museum in France. He was Felix Pauchet. Felix Pauchet was 60 at that time. He was one of the rationalist, atheist, and he's hardcore spontaneous generation theory supporter. And Pasteur was just 35 years old, and he asked Pasteur for a debate. So Pasteur did this experiment, and finally Pasteur was given a prize. He was accepted. There is this the religious climate at that time. The church felt that Pasteur and Tyndall's experiment proves what is the belief of the church at that time, because life comes from life. Life will not come from a non-living thing or any other source that is not related to the descendant. So finally he was given prizes, he was accepted, and Pauchet, he slowly left that debate and he went into some other researches, but he produced his own books in the same topic, and that it goes. And then later we know that Louis Pasteur was one of the founder of this germ theory of disease. What does this germ theory of disease that it says is that a disease is caused by some germ. You may not see it, but it is there in the air, it is there in the water, it is not visible, it is microscopic, but they are actually the causes of the diseases. And after him, Robert Koch, he was also a very important scientist, made great contributions to microbiology, not only microbiology, and vaccines. Many fields he did important contributions. He postulated 4 ideas that are considered as to establish a relationship between an organism and its disease. That condition, or that has to apply or obey to all these 4 conditions. What are those 4 conditions? 1, the abundant microorganisms in the infected, and that should not be there in the healthy people. If somebody is infected, or all those infected with some disease, the organism or the germ should be there in them. Because of the germ, they are getting ill. But in healthy people, they should not have this germ. That's the first rule. Second one, organism must be isolated from the infected host and grown in pure culture. So at this time, he was one of the a proponent of isolating microorganisms and growing them in pure culture. So this condition makes it is actually the agent that causes the disease. If it is the agent, then you have to be, then it must be able to be purified from the infected or diseased people, and it can be grown in a culture media. So that if you inoculate this pure culture into another person, healthy person, he will get sick, meaning that you are establishing a line of or connecting the dots, and you are establishing a relationship between a germ and its disease, and how it can be transmitted to another person. And the fourth one was the organism must be isolated, and it should be same original organism first isolated from the original diseased host. Now the story starts from here. A person infected, so he is having some diseases, and from him you should be able to isolate the organism and grow in culture. If this pure culture is injected into another healthy person, he should get infection, and after he develops the disease, if you are if you are isolating the organism from him again, then it should be the same one that you have isolated from the infected persons in the beginning. So it comes to a circle that it establishes that this is the organism and this is the disease that it causes. Because at that time, lots of diseases in Europe and in other parts of the world where people have no idea about or no clue about the causative agents. They don't know anything about because they don't see anything that comes in. They think everything is poison, or something happened to the earth. It produces poisonous gases, all that they believe. So these postulates very clearly establish the disease and causative agent relationship. But it was this is the beginning time. Even during the Robert Koch's time, he abandoned the first rule, because we know that not necessarily everybody who has some pathogens inside them are developing diseases. Even at this corona time, we know there are some asymptomatic people who are carrier, but they don't develop the disease fully. So, for example, we have E. coli, we have Salmonella typhi organisms in us, but we don't. Vibrio cholerae we have, but we don't develop diarrhea or we don't develop cholera disease. We don't develop typhoid immediately. We are just a carrier without symptoms. So from his time, he abandoned the first rule, but the other rules are there. But for viruses, again, until here, it's established only about the microorganisms of what now we say as fungi, bacteria, or anything, protists, something. But how do we introduce virus into this disease causative agent, purification, grown, and again infected into other person in this circle of establishing the causative agent and the disease cycle? Because viruses cannot be grown in pure culture. Why? Viruses are obligate parasites, but at that time it was not known. At that time, even there is no idea about what is the causative agent that now we call it as a virus. Because there at that time, even Hawk produced microscope, its resolution is up to only they can see some bacteria, that's all. Bacteria can be grown in cultural media. You give some nutrient in a liquid media, the bacteria will nicely grow there. But viruses, they failed in all these attempts when they tried to establish the causative agent disease and spread through these Robert Koch's postulates. They couldn't do it. Of course, you can collect some sample from blood or some pus from the infection. That's okay. That you can, if you can directly give it to another person, he will get the disease. But in between, you have to grow these organisms. You have to be able to show that they are all purely growing in some area. There they failed. Again, you confused them because viruses, they will not grow on their own in any media. They are obligate parasites, meaning they have a life only inside another cell. They infect a cell, we call them as their host cell, and inside the host cell, they grow. That's all. So, second point is, if you grow in some host cell or in some other host, after a few cycles of growth, of replication, mostly the viruses, they lose their virulence. So after you grow them, you inoculate them into another healthy person, the healthy person will not develop a full-blown disease. Again, one another point from Robert Koch's postulates is failed, because viruses, they attenuate or sometimes they lost their virulence when they are grown in cell culture or sometimes in a different host than their natural host. So that's the time. A lot of struggle to first prove that there are some organisms in the air that you cannot see, not that all that you see that grows are grown from spontaneously, they are generated. There is some germ that is in the air that you don't see. And then after proving all this, they had very hard time to understand what are all these agents that are still causing diseases, but not following, or you are not able to prove that through the postulates of Robert Koch. Now, at this time, Adolf Meyer, it's in the late 19th century, 18888, he observed that there is some deformity in the leaves of tobacco plants. Now we call them as tobacco mosaic disease, but at that time he just observed there's something happening in these tobaccos. But if you crush that infected tobaccos, take the extract and just spread it on another unhealthy leaves, that leaves will start get the infection, and they will get the disease, and they will also deform. He saw that, but he thought that probably it could be in bacteria, because there was knowledge about bacteria at that time. So he thought that it is bacteria. He just filtered that extract of leaf with a normal paper, and then he spread it on another viruses, another leaves, and they developed the disease. But so he simply thought it could be in bacteria. But he was the first one to observe that. within few years, his contemporary, a Russian scientist called Dmitry Ivanovsky, he used a Chamberlain filter. That's a filter. Chamberlain is a scientist. He developed this filter. It's made up of porcelain. They are very fine so that they can filter the bacteria, all bacteria at the time they know. So if you, so what Dmitry Ivanovsky tried, if it is a bacteria, then if you are filtering through this Chamberlain filter, you could be able to isolate them, grow them. But again, he failed. He couldn't get anything that causes the disease in the filter. So probably there is something, there is a toxin. He thought that it's not a bacteria itself, some kind of a toxin that is produced by a bacteria. That's why it is even smaller and it passes through the filter, produces diseases. So he again tried one more step, but he couldn't find out what it is. He just thought it could be a toxin. Then comes the another scientist, Martinus Beijerinck. He did also the same observation, but he thought that this is some kind of a contagium vivum fluidum. That means that it's a liquid or it's a fluid agent, an infectious liquid, it means. So virus is already a name in Latin that means poison. So he reintroduced the name poison for these agents. It's not bacteria, it's not toxin, it is something else, probably a poisonous liquid. So he gave the name virus to it. So that's how that's the virus, the name came into the picture. And even at that time, they don't know what is virus or anything. They just thought that it's a poisonous liquid from the extract. So it took some more time, or in the same year, Loeffler and Paul Frosch, these are all two students of former students of Robert Koch. They found the first animal virus. It's causing foot and mouth diseases in cattle. The causative agents are particle. They said that it's not a bacteria, it's not a toxin, it's not any poisonous liquid, but it's actually a particulate matter. It's so tiny, it can be retained by a finer filter of 0.2 micron size. So from there, actually, well, though Beijerinck gave the name virus, the real virus and the definition, or the basic definition for what a virus or some discovery about virus happened with this invention. Again, they can filter it, but they cannot grow it. We know the reason. They don't grow in media. That's the beginning. And then from that moment, there is enormous discoveries about our connections that connected the causative agent and the viruses. In all, 1901, they have yellow fever virus. 1903, rabies virus. Yellow fever virus was the first human virus that was discovered. Rabies virus next year, or in variola virus, 1908 polio virus, Rau-Sarcoma virus in 1911. Well, there are many other viruses within this time, but I'm just highlighting those viruses that cause serious diseases and human diseases. So I'm just highlighting only those. And then in 1915 to 17, because these 2 years, there are two different independent scientists. They isolated the bacteriophages. They found out what is that bacteriophage is. And then in 1933, influenza virus, and then there is a long discovery of enormous number of viruses. Finally, in 2020, we have something, a variant of SARS coronavirus. We call it as a SARS coronavirus 2. We have recently identified. We establish a disease and its causative agent. In all these discoveries, they didn't know whether the structure, they didn't know their genome or anything. It's just they are connecting a dot, two dots, our agent and the disease that it causes, nothing else. 1926, Thomas Milton, he first defined that viruses are obligate parasites. They are all particulate matters and they are all parasites. They don't have a life outside of a cell. One more interesting invention at this time that helped virology is indebted to this technique. That's the development of electron microscope. Ernest Ruska he developed electron microscope for his PhD thesis. So he, with his boss, They developed different electron microscopes. The 1931 was the first one, but it took 8 years to commercialize and to develop a fully, easily operatable electron microscope. They first saw a tobacco mosaic virus. TMV is tobacco mosaic virus. It was the first virus to be visualized with an electron microscope. It took 8 years after the invention of their electron microscope. Ruska's brother, he's a medical doctor. He could sense that how great this technology is. So there are many virus diseases that the condition, I mean, the indications are overlapping. The diseases are overlapping, symptoms are overlapping. For example, to differentiate between variola virus is nothing but the simple smallpox virus. So smallpox and varicella zoster virus. And he used electron microscope because Ruska is his brother. So 1948, first time for clinical differentiation and identification of two viruses, they used this variola virus and varicella zoster virus differentiation under the electron microscope. In the previous slide, I showed you colorful images of viruses. Actually, they are not colorful because viruses are too small. They are less than the wavelength of visible light. So they don't have any color for them. Usually, if you see under the electron microscope, they look like this. But in the softwares, you can give colors to them. There is advanced microscopic methods. You can scan, you can get more detailed views, and with the software, you can model them in 3D so that you are getting a nice view like this. Now, this is the tobacco mosaic virus. If it is in simple form, it is this colored one, and then high resolution 3D model based on scanning electron microscope and other crystallography details. With this, we have covered the story of how the virus discovered, and it is the history of virology also, the beginning of virology also. But viruses have a strong impact in the society. The spread of viruses, how it happened throughout the world. Today we have this, but how all these viruses are spread across the globe. That's the social history of viruses. We need to know that's how much viruses shaped our society, or how our society helped the virus to disseminate itself across the planet Earth. 80 million years ago, our ancestors of our modern human beings, they are infected with a virus, and that's herpes virus. So most of us, we have herpes virus genome in us. But there are other things. Viruses also emerge with its host, that is we. When we emerge, the viruses also emerge. The civilization is the reason for this, or what we call ourselves as society is emerging. We are civilized, we are growing in number, we are finding different ways. Before that, our ancestors, they were hunter and gatherers, they were living in a small group, they are not used to live in one area. There is no formation, there is no cultivation, agriculture was not invented. So they were just moving from place to place in smaller groups. So the virus, whatever emerges, it has to circulate within that group, and there is no chance for the virus to spread or to emerge again. So there are several reasons how our civilizational or our social activities made the virus to emerge. Number one is settlements. When people settled at one place, usually on the banks of some fertile river, they started cultivation, they settled in a big community, the community number increased. That's fantastic for a virus. If a virus has to emerge, if a virus has to spread, has to grow, evolve, then it should have a lot of people living in an area, a high host number they want. That's the case that's what we are trying to cut down the communication between people in this time, Corona time. So it was noted that the smallpox, 11,000 years is recent in the history of a virus. So smallpox emerged among the agricultural communities in India 11,000 years ago. But how this smallpox came suddenly from somewhere? No, it is descended as we are descended from some other our different type of ancestors in that group. So they descended from their poxviruses of rodents. From rodents, they descended and they crossed and they come into human being. They developed and they emerged as a smallpox virus. Around the same time, people settled in the plains of River Nile, Egypt. Mumps, rubella, polio, all deadly viruses, they emerged at the time. That's the time civilization slowly started building up. People started living in urban. They have lived in close areas. A lot of people are living in one area. They don't move here and there. And second thing is farming. Here, it's not important for the animal viruses, but for the plant viruses. In nature, in one area, no plants are there in its all. At the same time, within the same area, a lot of different plant species will be naturally grown. So if one virus infects one kind of a tree or a plant, it has less scope to jump, or it has less scope to spread to a wide number of plants, because the numbers are less. Immediately there is another different tree or a different plant. This virus cannot jump from here to there. That's the limiting force. But when we started farming, we started to cultivate a single plant in a huge area. Rice cultivation, paddy cultivation, maize cultivation, some potato cultivation, tomato, whatever it is, in the huge area, just only one plant is grown. That's fantastic time for emergence of different plant viruses. The plant viruses rapidly spread in that monoculture. For example, the SOBIMO virus, southern beans mosaic virus. So it's called SOBIMO. SOBIMO viruses, they emerged at this time in the Middle East. Third reason is domestication. People settled in one area, started farming. What they want, they need some animals for their health. Usually, we know that pigs, cattle, goats, sheep, horses, camel, cats, dogs, a wide variety of animals. We captured them, captivated them, we bred them in close to us. We started living close to them. That's the chance for zoonotic infections. There are viruses that infected these animals, and from them it started jumping, or it's called species cross species. It crosses one species, for example, from pig to human being. Or sometimes it is very interesting. For example, in the case of influenza, it started from duck, water duck, to pig, and then to human beings. So it's just started jumped to two different species, finally reached human beings. So it's rare, but we have a list of such infections or viruses that emerged and crossed the species, and from species from non-human or non-primates, they have come to human beings. It's a huge list. Influenza, the coronavirus that we know now, Ebola, Dengue, Congo, Lassa virus, Marburg virus, West Nile virus, Rift Nile, Zika. That's a huge list of. These are all deadly viruses. Most of these, West Nile, Ebola, Dengue, they all cause hemorrhagic fever. They are deadly. Okay, most of these viruses, they jump from species to human beings. Third reason. And then fourth reason is war. So in the society, I'm going to give just one or two examples. There are lots of notes spread in the history of human civilization, where there are so many wars. Wars not only brought death to people who fought, but also death to people who are not part of that, just got infections, and when they are turning to their native place, they're just spreading this. One best example is there was a war. There was a smallpox epidemic that happened in the 2nd century, 65 to 180, through the Roman soldiers. They went on a war in Seleucia, it's today's Iraq, and on the bank of the Tigris River. That's this area. So Roman soldiers were indulged in a war. So when they are turning back to their native place, Roman kingdom, they also, at that time, unfortunately, there was this smallpox epidemic in Seleucia. They contracted it, and when they come back, all the way they spread the disease. Within few years, 5 million people in the Roman Empire dead. And then there was a long time with these crusades and Islamic conquests. They are all very important reason for smallpox spread. I'm again giving smallpox with, this is very important disease. If you calculate all the smallpox deaths happened in all these 1,000 years across the globe, from Europe and in Asia, in other continents. That's really huge. Every time it took millions of life, not one or two, but then collectively smallpox was a deadly disease. Everyone was afraid of that, or they were more care about that. It's the frequent epidemics in Europe between 5th and century. That's the time that these crusades and Islamic conquests happened, and the people are going from one area to another area for spreading religion or to claim some space, and they were indulged in war. There will be some epidemics, and they brought these diseases back to their place. And between these 2 centuries, there are multiple incidents where smallpox spread because of the war. In 13th century, we know that Genghis Khan, some Mongolian emperor, he invaded Europe, and he actually spread, or his invasions, there are 3 time points, they say, in the 13th century. In all the 3 invasions, the measles got spread. A pandemic of measles was there in the Europe. Another important reason is colonization. And from 15th to 18th century, European colonies spread smallpox, measles, influenza into Americas and Australia. Before their arrival, before this European colonists' arrival into these 2 continents, Australia and America, they had no smallpox, no measles, no influenza. These are all the deadliest diseases at that time. But the colonists, they're aspiring to grab a land and to control the people over, dominate them, make a land for them. But eventually, they spread the diseases. The interesting or the most sad thing is, within 150 years of Columbus's arrival, from Spain to Columbus's arrival in Americas, the Native American population is reduced by 80% because of these diseases. They introduce all these diseases. So that's the impact of viruses. We know from the previous slides that the history of viruses starts from somewhere in the 17th century. They started to think that it's a bacteria, toxic fluid or something. But just, is that the beginning of that people are observing something happening abnormally? No. There are proofs that people observed conditions or diseases caused by virus infections. For example, if you notice this, it's a tablet from the Egypt, almost 3,500 years before. There, a priest is depicted with a very classic, or exact this hanging foot or drop foot, or this deformed foot infection by polio infection, meaning that they have noticed, they don't know what it is, but they know that this is abnormal. So in some way, it has been registered either knowingly or unknowingly. There are interesting things. For example, Mesopotamian laws says about rabid dogs, how they should be controlled. If a dog of owner gets rabies, how it should be controlled. And there are many things written about rabid dogs. Well, rabies has also, the name rabies comes from the word, a Sanskrit word, rabas. It means rage, get angry about that. So other things, variolation, for example, this smallpox vaccination in India and China and in Turkey, the people already know about how to protect themselves from this deadly disease, smallpox. In the 11th century, for example, one lady, she's called Lady Montague. She was the wife of an ambassador of Britain to the Turkey. So she lived with her husband in Turkey. There she saw people are doing this practice. They're just making a prick in the pus of a person infected with smallpox, and they are just scratching on the skin of healthy person. He develops a mild disease, but then he is fully protected from the full-blown disease. So she noted it, and she went back to UK. She talked to the government. She made her doctors to do that. Actually, her brother died of smallpox. So she wanted her son, 5-year-old son, and when she was there in Turkey at that time, so she wanted to save her son. She asked his embassy doctor to do that. Oh, that's how this variolation or poxyvirus vaccination, well, there was no real vaccination known at that time, but this practice, we had it from this Asian countries, China, India, from Turkey, through the Turkey, it went to the Europe. It was a lucrative business for a time. They charged even at that time 5 to 10 pounds for variolation. Government made the variolation to protect the people as a mandatory. Many people were against that vaccination. They didn't want, there was an uprising against the government. Finally, the government had to give up the idea of compulsory vaccination. It all happened 1,000 years back. So with this, we understand the story behind the discovery of viruses and the social history of viruses. And then it's that all. No, we know that these are all the agents. We know that they can be seen under the electron microscope. We know that they are obligate parasites. Is that everything over? No. There is two important controversies about the viruses. First one is their origin. Okay, we know there are viruses, but where do they come from? We have microbes, plants, animals, different types of organisms. We don't know, but we have some similarities from all this. But where does this virus come from? There are three. Of course, there are even more interesting hypotheses coming up, but these are all the classic three hypotheses that says or speaks about try to answer the origin of viruses. The first one is called the progressive or escape hypothesis. It says that fragments of genetic material, for example, we all have some a portion of our DNA that's called retrotransposons. They behave exactly like a virus. For example, all these retroviruses, including HIV, this HIV virus, its genome, and what we have in us, that's called the retrotransposon, they look almost same. Of our retrotransposons, it misses some of the important genes of the viruses, that's all. But they look, and the way they behave is almost similar. So this hypothesis says that these retrotransposons are very funny. They can, by their own, copy their gene and paste it in a different area of our genome. Or they can cut their genome from one place, themselves from one place, and paste it into different area of our genome. There are several diseases that cause because of this random intrusion of this gene into different areas of our genome. We saw that these elements, they escaped from the host cell, from our cells or from any animal cells, plant cells, whatever this is, because all eukaryotes have some retrotransposons. In fact, 50% of our genome is just retrotransposons. So this hypothesis says that these mobile genetic elements, they escaped from the host. Somehow they got some proteins under covering, wrapping, and they become viruses. Plasmids are also an example of this. But here there is a catch. According to this theory, these are all the retrotransposons. They can jump from one area to another area. If this escape from here and become a virus, then the opposite is also possible. We can ask this question, how do we have this retrotransposon, first of all, in our genome? Then there is the answer, probably these viruses were originally there. These retroviruses infected human beings. The genomes integrated into our genome, then they stayed forever, that's all. So there is a catch here, but it's not answered by the progressive or escape hypothesis. So there are other theories. The second one is opposite, it's regressive or devolution hypothesis. Here it says that the viruses are remnants of cellular organisms. The best examples are NCLDV viruses. It does mean nucleocytoplasmic large DNA viruses. There are viruses. For example, this smallpox virus, there are giant viruses called Mimi viruses. These viruses, they have so much of genes. A normal virus, they have very few genes. But these viruses, they have a lot of genes, a lot of proteins. Why do they have so much of proteins, a big genome? They are, in fact, able to produce their protein or transcribe their gene, not in the nucleus, but in the cytoplasm itself. they have the accessory proteins to achieve this. There they are saying this interesting idea that these are all the viruses that once lived within the cells. The relationship between one eukaryotic cell and this cell started as a symbiotic relationship, and after some time, this intruded by a cell, they become more and more dependent on the other cell. When it gets everything from the other cell, it lost it lost most of its genes that are necessary for it, because it is unnecessary now. And now after some time, because it is now is different and it is dependent on this, it emerged as a virus, and then this virus, whenever it wants, it can infect these cells and replicate them. They are not dependent on the cell anymore. They come out and then go to another cell. That's how they are. These viruses have developed. That's the regressive or devolution hypothesis. The first two hypotheses they say about the viruses, or they assume that the cellular life form is already present, either from which it came out or from outside the viruses came inside. What if there is an alternative idea that says that the viruses are the first one? Before all these bacteria, archibacteria, or eukaryotic cells, everything went. What if these viruses are the one that are the origin of all these life forms that you see now? Yes, very interesting supportive evidences for this theory also. And this the third one is now getting more and more evidences, proofs. It's very interesting, because viruses have evolved from a self-replicating material. There is a concept called RNA world, prebiotic RNA world. Before any cellular life form existed, no bacteria, no plant cell, animal cell, anything, but there was just simple naked RNA. That naked RNA was able to replicate by itself. That naked RNA can do all its own job for itself. That's all. So from that RNA, the first form that existed or evolved was the RNA or viruses. From the RNA viruses, DNA viruses, and all these life forms evolved from that. So there is this fantastic proof of ribozymes. We still today have ribozymes. Ribozymes are RNA molecules, but they are able, they have the catalytic ability, enzymatic ability. They can do some enzymatic job. It's not just an RNA piece, but it's act as an enzyme also. That's the best proof for the virus first hypothesis, that puts virus is the beginning of all life forms. From viruses, now you have all these three domains of life, what people call. Interesting. The theory says that life or viruses could be the origin of all these three domains of life, that is called as bacteria, archaea, and eukarya. Based on this, they all started from one point, and that's called LUCA, the last universal cellular ancestor. It's a hypothetical cell or a life form. Nobody knows whether it existed or not, but it's theoretically put that all these different life forms should have started from something. That something is known as the last universal cellular ancestor, and that's it. Funny thing is, the hypothesis say that the viruses are there somewhere in the beginning, but viruses does not belong to any of these three domains of life what we say today. They are not even the LUCA. Then what viruses are? Because why viruses has no place in the three domains that we have today? There are a lot of virus proteins, but these proteins, most of the proteins, viral proteins, have no homologous in modern cells, any of these cells. Virus genes originated in the virosphere during replication of viral genomes and or were recruited from cell lineages which are now extinct, meaning that these viruses, it was a virosphere at that time, just only viruses. These viruses probably had some cells to host them. Now these cells are not existing. They are not here. So the viruses, the host, the proteins are not there in any of other life forms. So it was a different sphere. It doesn't belong to any of these. So the origin is controversial. There is no place for the virus in any of the domains of life that we have today. And also the viruses are omnivalent. But again, with having all this ability to do variety of things, they're still outliers. They don't belong to any of these three domains of life. Imagine that all life form, as we know, it's all containing DNA as their genetic material. DNA is the brain of all cells, either bacteria, archaebacteria, plant cell, animal cell, yeast, anything you take. Viruses are very different in this. The genetic material of a virus can be DNA or RNA. Probably a DNA, some viruses, they have a short segment of RNA within the DNA. There are viruses with DNA or RNA with some proteins attached to it. That's the genetic material for them. If it is a single-stranded DNA or RNA, it can be plus strand, minus strand, or ambisense, meaning plus strand means you can, for example, if it is a plus strand RNA virus, the protein can be directly produced from it. If it's a minus strand, the minus strand should produce a plus strand. From there, it produces the proteins. Ambisense means one strand of RNA or DNA, it contains half of it is plus and half of it is minus, like this. No way that no wild guess somebody can have such a nucleic or a genome can exist as a base for some life form. It can be double-stranded, it can be linear, or it can be a circular molecule. Not necessarily it should be a one piece of genetic material. Some viruses do, they have 10 pieces of genetic material. It's not just one single strand, it's just bits and pieces of, but all the 10 segments collectively code for all the things that the viruses need to survive. It can be gapped. For example, there can be a double strand and there can be just a single strand that connects these double strands. Hepatitis virus is the best example for that. So they're omnivalent. They have all the possible method of encoding genetic information in nucleic acid. Again, they are outliers. They are not belonging to any of these. Why? Interestingly, what we think as viruses from this discovery, we know that viruses are notoriously small. For example, the most smallest virus is a porcine circovirus. It's just 17 nanometer. Parvoviruses comes close to it. It's 18 to 22 nanometer. That's all. But there are different life forms of viruses. It's very interesting to know because these informations are very recent, from 2004 to 14. That's all. This giant virus is called Mimi virus. It is 600 nanometer in size. Imagine a normal virus of 100 nanometer. This is 6 times bigger than any normal viruses. It was isolated from a cooling tower in UK, I think, or in France. And then after a few years, 1911 or 2011, I think, they discovered another big viruses. They named it Pandora virus. It's even bigger. It's 1 micron in length, almost like a bacteria, some small bacteria. It's 0.5 micron in diameter, in width. Recently, in 2014, from the Siberian area, from the deep of ice, they recovered this virus. It's called a pithovirus. It's even bigger. It's 1.5 micron in length, 0.5 micron in width. These pithoviruses, interestingly, millions of years, or we don't know how many millions of years they are there inside this ice pile in the Siberia. The moment they took and they infected these collections of ice crystals in amoeba, that amoebas are killed because these giant viruses usually kills or lives within the amoeba. So they found the interesting thing is some life form that can withstand, that can be in a dormant state for so many thousand or million years. And again, when comes time favorable for them, they are coming with their full infectious ability. Luckily, these viruses, they don't infect human beings, but that's posed a serious question. What if the snow melts in the polar region? All those fossils of viruses that are residing, they will come to alive, they will come back. We don't know what kind of virus diseases our ancestors of our modern human being they suffered. Or what if these viruses come back to life and start to attack us? It's a nightmare. virus, the biggest virus, the smallest bacteria is just 200 to 300 nanometer. A virus is 5 times bigger than that. The smallest eukaryotic cell is a portion of it, algae. It's 800 nanometer. A virus is bigger than a eukaryotic unicellular organism. That's great news. These are all the new informations from the virology side. See, this is a bacteriophage that infects a bacteria. Bacteriophage is a virus, we know, that infects the bacteria. See how a bacteria looks a giant in front of a virus. The other picture shows that these are all the bacteria. When they are close to a eukaryotic cell, these are all human colony cells. Imagine that how much they are dwarfed by our cells. Imagine the sizes of the virus, compare from so tiny virus to a bacteria, and how a bacteria is nothing in front of an eukaryotic cell. The second one is a controversial existence of the virus. What do you mean by existence? What I mean by existence, their nature, whether they are alive or not, what people think. No, there is no doubt about a bacteria. It's a living thing. But viruses, can people accept that it's a living thing? No, not many people accept that. For example, that the definition about life itself is highly different. NASA simply says that whatever it defines life as what is able to self-replicate itself and evolve over time. That's okay. Viruses do that. They replicate themselves, self-replicating. And we also know that we get some infection, the viruses grows and replicates in number, and HIV is an example. We see them in before our eyes, coronavirus are example. They are evolving from one stage to another stage. That doesn't mean that they are alive. But on the other side, biological definition for life is they should have some metabolic processes, they should produce energy, they should be to some extent self-reliant. But viruses, they don't have any proteins for any metabolic processes, meaning they don't produce any energy for themselves. They don't have any ribosomes. Ribosomes are the one that makes the proteins. So viruses, of course, they have many proteins in them. None of these proteins can be produced by themselves. They have to hijack from the host cell. So what does it mean? That they don't have anything that to claim that it is a life form. They don't do any physiological process, metabolic processes, meaning that they are alive. It's a big question. The opinions are divided into two, or probably they are living in or the border between chemistry and life, because a virus can be explained as some people say that they are like a chemical sac. They have some nucleic acid, some proteins coated by some proteins. They have some lipids if it is an envelope, virus like a corona or HIV, but they have no life, just chemicals. But the moment they meet their host cell, they suddenly activate themselves and actively involving in replication, producing proteins, and come to a life. So probably they are between chemistry and life. They are just in the border, or probably we need to redefine what is life. There's a definition that says, how could viruses be defined? They are kind of a borrowed life. They don't have life of their own. It's just a borrowed life. Again, we have to redefine life if we want to say that HIVs are, the viruses are life. It's up to us. Each and everyone can argue with the knowledge about viruses, how they do they manage many things, whether they are alive or not. You can think whether they are just a hijacker. They get into some place and then hijacks everything, kill the host. You can call it a hijacker, or you can think that they are like a smart traveler. They don't carry much things. They go very simply, get the job done, replicate into many. They just go to the next host. They're just very smart. They just don't carry much things they need, but they are managing their life. They are surviving. They are evolving. They are successfully increasing their progeny. Smart. Now comes to the title. It's yin and yang. In Taoist metaphysics, in the philosophy, we normally have these dichotomies or dual things: good, bad, light, dark, femininity, masculinity. All these dualities that says that opposite of that. But these Taoist metaphysics, yin and yang, it defines all or complementing each each other, and they are indivisible whole. Within the white, you have a black dot, meaning that within the yang, you have a yin. Within all light, there is some darkness. Within all the darkness, there is some light. Within all the good, there is some bad. Within all the bad, there is something good. So it's that what we judge are just our perceptions. That's not real. The duality is indivisible. It's just one. So based on this philosophical background, how can we classify the viruses now? Are they good? They help us, our friend, or they are going to wipe our human race, or they are our foe? They are disturbing us. It's the thing. Well, the virology started from the point when we were care about ourselves, our plants, our cattle's, livestock. When there is a disease, we start to search what is happening in them, and then we found out the viruses. Now, on the opposite side, when we want to know more about the viruses, we started to learn about ourselves. Most of the information that we know, the molecular biology, as we call it now, all those informations, thanks to the viruses, because we were trying to understand the viruses, how they are replicated in the cell, then serendipity, it's luckily that we came to know more about ourselves, the mechanisms that govern our normal cellular metabolism on everything. For example, a study of small DNA viruses led to the discovery of promoters of eukaryotic RNA polymerases. We know that how important a promoter is. We came to know only when we started studying our DNA viruses. Before that, we don't know that. Study of cancer-producing viruses led to the discovery of many cellular oncogenes. We have oncogenes that can cause cancer, but we didn't know that they exist in us. When we started studying about these viruses that cause cancer, then we came to know about, yeah, we have these genes already in us. That's RNA splicing. We didn't know that RNA, there is something called as splicing happens. There is this big RNA, there is information nonsense, and these nonsense are clear. The information is made in one clear unit. We didn't know that's something happening in us. But only after started studying the mRNA from DNA viruses, we learned that it is happening in the virus, and the virus is doing all that with our eukaryotic cellular machinery, that it is happening in us. Understanding of cellular DNA replication was facilitated by studying phages and DNA. These are all foundations of molecular biology. Most of them are, we came to know about these informations only after we started learning the viruses. We started our focus to understand what viruses are. Now, in few slides, I will complete this. I'm sorry for that. Now, with the yin-yang basics, now I'm going to show you a few slides, and we can decide whether it is friend or foe or yin and yang. There's an estimation that 10 power 13 viruses on Earth. Sorry, it's 39. Most of these viruses are bacteriophages, meaning that they kill the bacteria. And among most of the bacteriophages, most of them live in the oceans. They are there in the oceans. What does it mean? Or how it is helpful for us? The other information is, in oceans, more than 90% of the biomass of the ocean is just microorganisms. Everything, bacteria, fungus, algae, everything. If 90% of the biomass is oceans, and you know the way speed, how these microorganisms can replicate, how it could be there dominating the oceans. But here is our hero, the viruses. They kill approximately 20% of this biomass every day. So they are the checkers. They maintain the stasis, the homeostasis of the biosphere of the oceans every day. Without these viruses, the algal blooms completely covered, and they will harm all other ocean living lives. So there are 15 times more viruses in the ocean than bacteria and archaea. That's how they are able to clear this biomass every day. They are doing a fantastic job. If the viruses are not rapidly destroying all this biomass produced every day, the algal bloom will be very harmful for and kill all other marine life. Also, it helps maintaining in the ecological balance of different blue-green algae, so that these blue-green algae are there. What it does, it's a fantastic job. They are producing adequate oxygen. and then it helps our earth, the life on earth. We call it this, like, bacteria are having no life, but they are actually supporting all the life on the earth by doing this. Here in this picture, you see that a bacterial cell is lysed, and small dots here, these are all macrophages. So, sorry, bacteriophages, they are all coming out of cell. One bacteriophage infected a bacteria, replicated into hundreds, kill the cell, and coming out of it. What does it say? That bacteriophages can simply kill bacteria. What if it can kill the pathogenic bacteria in us? That's the idea. I forgot to mention his name, Felix de Herel. The identification of bacteriophage happened in 1915, around 1917. But even 20 years before that, there was a British biologist. His name is Hannick. He was there in India. He noted the culture of Indian people taking or claiming it as a holy bath of taking a bath in the Ganges. He analyzed the samples or water sample from Ganges. He found that the Ganges water, the Ganga water, is killing the Vibrio cholerae. He even did some small experiment in people where they have cholera infection. For one set of people, he gave the Ganga water directly, and for other people, he gave the Ganga water boiled and gave it. Those who drank, who take the Ganga water fresh, they all rescued from, or they are all rescued from the cholera infection. But those who got the boiled Ganga water, they got sick. He said, there is something in this Ganga water, but it is thermosensitive. If you heat it, that magic thing goes out. So he did some experiments, he proved and he showed all this, but he has no idea what is there. It took 20 years for two scientists to understand it, Frederick and the Felix. They did this research and they found out, not from the Ganga water, they collected sample from somewhere else. They found that these bacteria are killed by something, and they found out these are all the bacteria eating creatures. Why finally they know it is a virus, they named it as a bacteriophagus. So after this discovery, Felix got really popular. He traveled and treated, he traveled to many countries. Wherever there is a sudden outbreak of some infectious disease, he would go there, he would prepare some cocktail of bacteriophages, give it to people, and he cured many people, and he was doing this. And there is this scientist from Georgia, his name is Georgi Eliava. They met, Felix and George Eliava met. George Eliava, he invited Felix to establish a bacteriophage producing facility, or to visit his lab and do some work on bacteriophages. So Felix went to Georgia. They established this phage therapy in this Eliava Institute in Georgia in 1934. He just stayed 1.5 years, or sometimes that's all, maximum 2 years there. But at that time, there was this Finnish-Russian war was happening. Georgia was essentially part of Russia at that time, or in the close connections. The Russian war, the Finnish war, Felix treated almost 18,000 soldiers with bacterial or bacteriophage cocktails against anthrax. He first isolated the bacteriophages specific for anthrax, and then he gave it to the people, especially soldiers. You know how serious this anthrax is. So most of the, with 80% success rate, he treated the soldiers, so they are safe from amputing organs, leg or something. Okay. Now, how can this be helpful? We have this problem of MDR here is multi-drug resistance bacteria. There are lots of bacteria, especially these pathogenic bacteria, because of the continuous exposure to different kinds of antibiotics. They got some mutations, they are evolving, getting resistance more to all these bacteria, sorry, all these drugs, not for one drug. Some bacteria, they are resistant to many different drugs. These are the problematic bacteria now. There is no drug to treat them. So what if that you are reviving an idea of using bacteriophages to kill these? Originally in 1960s, the Russians were very much interested in, they conduct a lot of trials, but they didn't publish it, and later they published the results. But now these strains, or these antibacterial resistant bacterial strains are reviving the idea of developing serious phage therapy to overcome these antibiotics, because we don't have any new antibiotics. Just two new classes of antibiotics we have developed in this 30 years. We don't have anything new. So there should be some alternative source to tackle this problem, and the hope is on the viruses, the bacteriophages. Evolution, sorry, evolution. Evolution can be shaped by viruses? There are lots of very interesting evidences that proves that how evolution as a whole, not only evolution of something, it's all evolution of bacteria, evolution of plants, animals, human beings, including human beings, all that is shaped by viruses. That's a great thing. Well, let's just see a few examples. We spoke about the retrotransposons. These are all some bacteria, retroviruses. They infected us, they become part of us. After so many millions of years, they lost most of their virulent genes, and they have only a basic structure of their original gene, and they are not toxic anymore, they are not infectious anymore, but they are trapped within our genome. I told you that there are almost 45 to 50% of our genome is containing these viral origins. So we are all half viruses already. 47 human genes we know now we have. They are all derived from these retro elements. It's meaning that these genes were not evolved. It's not like the usual Darwinian way of evolution. It's just these viruses directly donating some genes for us. But it takes a lot of time, and there is this grand natural experiment. It finally takes 45 genes out of all these infections, and now they are helping us. Or there are views that see in the opposite way. I will give you some best examples of how these viruses they are helping us. There is this gene called syncytin. There are two genes that we have. These genes, syncytin, are expressed only in the placenta of human beings. It fuses the placental cells together. That's called a syncytium, because the viruses, they have this native ability of syncing two cells, host cells, and creating a giant cell. So these syncytin, it creates a fantastic placental cell fuse that makes like a barrier. These genes are present in our placenta. Those two genes, they have their origin from the viruses. Also, these helps the fetus from mother immune system. Otherwise, the mother immune system will attack the growing fetus. Sometimes it protects them. Not only we have, there are other animals, other large primates, other animals like mice, cats, dogs. They have, but they are not derived from viral syncytin genes. So we have got these syncytin genes. They are doing a fantastic job of protecting the fetus. We got as a gift from the viruses. They are now fantastically working for us. Interestingly, pigs and horses, they don't have a layer of fused cell in their placenta, no virally derived syncytin genes. Sorry, mice, cats, they do have, they have also viral derived syncytin genes, but not the one from the human beings. But pigs and horses, they don't have any this fused cell forming in their placenta. That's theirs is entirely different. Probably their pigs and horses ancestors, they were not infected by any retroviruses, so they don't have these syncytin genes. It's a gift for only few animals, including human beings. One retrovirus infected human beings some 45 to 60 million years ago, so long back. And these become endogenous, meaning that these retrovirus started to live within us. They are doing a fantastic job. They switches, meaning that they switch on that one important gene called AIM2. These AIM2 genes, upon what? When there is a cell and it is infected by a virus, immediately this AIM2 is signaled. Who is signaled? Who is regulating? Those retroviral genome parts that we have got in our genome. What happens to that infected cell? The infected cell immediately commits suicide, because it has already gotten virus infection. If it allows the virus to grow, hundreds of viruses will come infect other cells. What if immediately dies itself? Then there will be no virus spread. Some people, funny, they say that it's a kind of a double agent. There are many virus proteins. They are originally from virus, now part of us. They are now helping us to fight against other viruses. Another gene, it's this, it's pro-DH, it's proline dehydrogenase gene that is expressed in the brain, especially in the hippocampal region. This is activated by a retroviral insert. They have an enhancer sequence in that gene. It's called human-specific endogenous retroviral sequence. And this retrovirus derived sequence is the one that regulates this pro-DH or pro gene. This pro gene is very important. If there is a fault in the proper expression of this, it leads to many neurodegenerative diseases. So they think that this interaction of enterovirus or retrovirus genes and the pro-DH, they have contributed to the development of our human central nervous system itself. So much contribution for us in the evolution by the viruses. Genes in the cells that build our face, it's interesting. We have a flat face. We have our close primates. They have a different face. Well, when we are growing in the fetus in our mother's womb, those cells that finally make your face, they have some genes that to express. The genes are all controlled by human-specific endogenous retrovirus elements. That's why we have a flat face. Our close relatives, these chimps, they don't have a flat face. They have an elongated face. So the identity of our face is a gift from the retroviral elements, virus infections that is living inside us now. More than 300 different crab. This is very interesting. These are all crab zinc finger proteins, meaning that these virus infect us. They leave the traces of their gene in us, and they are doing something for their own. No. If it is allowed to do everything on its own, that most likely will end in disease conditions. The recent discovery from a group of scientists, they say that these crab zinc finger proteins are the one. It's like they use this beautiful word called, they are tamming the incoming viruses. They are acquiring the virus, they are tamming them, they domesticate them, they delete some part, or they make them silent, and finally exploit the good things in the virus genomes in us. So that's why the virally derived DNA, they are all these examples that we've seen, they are all helpful for us. But how they are helpful by their own? No, they say that there is something from our side that it tams, domesticates these incoming viruses and make all this possible. Well, we have seen so many examples from the retroviruses. HIV is also a retrovirus, and it's a big problem now for the past 40 years from 1980, at least from what we know. Meaning, like we have all these retroviral transposon or endogenous retrovirus elements in us that is helpful for us. Can the current infection of HIV will also be endogenized, meaning that this gene will also be made as part of us, and then we will be immune against HIVs forever? Theoretically, it is possible. For anything that has to be endogenized to be made into our own, it has to happen in the sperm cells or in the egg cells. HIVs has already been proved that they can infect the spermatozoa. So if these genes are there, sperm fuses with an egg, and that will be there forever. So maybe it may take thousands of years or millions of years. After that, HIV probably, theoretically, yes, it is possible that HIV genes will part of us and we will be against the HIV. I mean, resistance to HIV. A lot of viruses, not only retroviruses, there are different DNA viruses, Bournaviridae, Filoviridae, Rhabdoviridae. Most of us, they are there in us. So that's what I said. There's these viruses, not only retroviruses, different viruses, they have left the remnants or traces in us, and that's we are almost half viruses. There is one very interesting idea. It's called all this super infection exclusion, meaning if one virus infects one cell, the virus will protect that cell from the same virus or a closely related virus. No other virus is closely related virus or the same type of virus can again infect the virus, this cell. This being, now this is very interesting. Koala, you know these animals, koalas. They say that it is now live happening in the koalas because there are koalas that caught from some part of Australia. They don't have this. They were trapped, caught from some 100 years ago, and their generations, they don't have this KORB, means koala retroviruses. They don't have this koala retrovirus genome in them, meaning that within this 100 years or just above this 100 years, this koala retrovirus endogenization has started being. This confers protection for the koalas. Those koalas that have this endogenous koala retrovirus genome inside them, they are all mostly resistant, not mostly, almost. They are all resistant to exogenous or any koala virus from external source. They are completely perfectly right. Those koala viruses where they don't have an endogenous retrovirus inside them, they are all susceptible to the virus and they die. So this is right now happening. It will take time for all koala viruses, all the koala viruses, they die and then they get a real protection. It will take time. Endogenous BDV sequences, this is another example. This is very interesting. HGT, horizontal gene transfer by virus. Beyond Darwin, what does it mean? Beyond Darwin, it's not the way that Darwin probably expected that evolution would happen. It's just a response for the nature, and the nature selects, isn't it? Well, here the horizontal gene transfer happened by the virus. We have seen so many examples. The virus has give some gene to some animals, plants, bacteria, and they are endogenized. They got this gain of gene and the function, and that gain of gene evolves that species, a human being, bacteria, whatever it is, into another level. So earlier I showed that there is this tree of life. It started from some common ancestor, and these 3 domains of life. It's not anymore that people think that they are diverging from their lineage. Actually, all these 3 different domains, of course, virus has no space in these 3 domains, but virus infects a bacteria, gives this gene into another bacteria, or a virus gives its gene into some eukaryotic cell. From archaebacteria, virus infects across the species to another bacteria. So actually, the whole evolution, there is a whole lot of contribution by the viruses. They are capturing genes from one and transferring it to another. That's why these evolving new cells, cellular life forms are gaining new functions. That's the thing. Okay, 3 more minutes, I will finish it with 8:40. 2 slides. We know that viruses cause diseases. How can we use the same viruses to cure them? That's the idea behind the viral vectors. The idea is simple. You use a virus because we know that viruses are very clever in infecting a cell, transferring its gene or its DNA into the cell, and then do everything. What if that you break the leg of the virus, meaning that the virus is intact in the beginning? You insert any gene that you want to correct, or as like a therapeutic gene, use this virus to infect a host. When the virus infects, you have already removed most of the essential gene for the virus, so the virus cannot replicate, but your job is done. You have sent the gene that you wanted to send inside the host, a human being, animal, plant, whatever. So this gene therapy, gene transfer, viruses are naturally doing it. We see that in HGT, horizontal gene transfer, they are doing. So what if that you are breaking only the chain that how the virus replicates? Otherwise, the virus can be used as the vector, fantastic vector for gene therapy. That's a fantastic idea. And there are a lot of clinical trials for different diseases already going on. Last slide here, the killer turn creator. Cancer, tumor, this is many reasons are there. What immune system is sometimes is the reason for creating cancer. There are carcinogenic chemicals that causes cancer. Radiations can cause cancer. Viruses can also cause cancer, but no viruses except only one epsilon virus or something. Yes, yeah, epsilon virus. No virus has an intention to create a cell into a cancerous cell, but it's a side effect. They don't need it. They don't need to transform a normal cell into a cancerous cell for their survival, but it happens. It's a collateral damage. That's all. But the viruses, we know that they are infecting a cell and transferring into that. Now, there is a fantastic idea using the same viruses, some viruses, to kill the cancer cells. You infect the cells with viruses. They will not infect normal cell, so that you design how beautifully that the virus will recognize only a cancerous cell and infect and kills it. And then that's called the oncolytic virotherapy. Viruses are one of the reasons for creating a cancer. Now we are turning it the killer to kill the cancerous cells. There are ways how we can do it, achieve it. There is already one immunologic, a genetically engineered herpes simplex virus. It's the first oncolytic virotherapy that's approved in US and EU, Europe for local or advanced or non-resectable melanoma to cure. So with all these informations, we have covered both sides: infections, the pandemics that kills lots of people, the problems that virus creates to the livestock, to plants, and then the other side, the young, where it is helpful. We are trying to use the same virus. Even HIV is used as a viral vector to cure many diseases, to send genes into the cells. So I leave it to the listeners to decide whether it is yin and yang, or you side with our friends or their enemy, or you side with their non-living stuff or their living creatures. Everything up to us. Thank you for listening, and I'm sorry for taking much time. Thank you.